Okay, so this chapter is about microbial metabolism. So metabolism, it is the chemical reactions that happens all at once in a specific organism. So typically it is used to build up macromolecules like lipids, carbohydrates, nucleic acids, and proteins. But also can be used to break down the nutrients so that it can sustain life. And then that happens in every living organism, including microbes. Now, sometimes microorganisms can live with their metabolism certain wastes that can cause some signs and symptoms in the host. However, most of the times, or they can produce food spoilage, but most of the times this metabolism is not affecting the host or the environment. So here it is, the big picture of what metabolism entails. So it includes this glycolysis cycle in which, or pathway, in which you have a glucose molecule entering, and then it's broken down into pyruvic acid, which later on is going to be converted into acetyl-CoA, and then it's going to lead to ATP, along with a little bit of ATP in this glycolysis cycle through oxidative phosphorylation. And then we have as well this pentose phosphate pathway that is going to lead to the formation of nucleic acids, which are, of course, going to form RNA and RNA and DNA. And then we're going to have lipids that can be oxidized, and then you have glycerol molecules that are going to be useful for storage of energy or either to get energy. Now, proteins basically are links or chains of amino acids in which you have enzymes that links them together, like peptidases. And then these amino acids that are linked together will form a specific protein, can be functional or can be structural. Name of, for instance, or a classification of a functional enzyme will be, or proteins will be enzymes. And then you will have all of these four major molecules that enters into the metabolism. Sometimes some of them, they can be interchanged, like in gluconeogenesis, you can have formation of glucose out of amino acids, or out of lipids, you can form these glucose molecules. And then some organisms can have photosynthesis. So that means that they grab light, and then they convert that light together with CO2 and water into carbohydrates and, and oxygen. So, catabolism, it is the breaking down of large molecules or complex molecules into smaller ones. And it helps us to obtain energy. So most of the catabolic reactions happens through hydrolysis, which is basically adding water so that you can break down the chemical bonds between the molecules. And this is an exergonic reaction, meaning that you release energy. Now, in anabolism, which is the opposite of catabolism, you will have the opposite. So basically, you will have small molecules, bring them together, link them chemically, and then you will form a large molecule. This is known as a biosynthetic set of reactions that are energonic, or they use energy. And typically, you will dehydrate or condense these molecules because you can remove out of one monomer, remove a hydroxyl group, and then from the other one, you will remove a hydrogen group, and then, a hydrogen molecule, and then you will end up with water. So here is an example for this formation of ATP and the breakdown of ATP. So ATP, or adenosine triphosphate, it is formed when you have adenosine diphosphate, and then you add inorganic phosphate plus energy, you form ATP. Now, ATP can be broken down into adenosine diphosphate, and then you will have inorganic phosphate free, and then you have energy. So both of them are coupled, these two reactions. And this is the way it's illustrated. So an example would be in the catabolism of glucose, where you have to add ATP. So you will have, as a byproduct, CO2 and hydrogen, but at the same time as you are adding ATP during glycolysis, you will have 2 molecules of ATP generated. So it's an investment. And then another example will be when you are having amino acids, free amino acids, you can link them, and then you can form a protein. And you have to use ATP, and then you break it down into adenosine diphosphate and into inorganic phosphate. Metabolic pathways in general are sequenced. They have molecules, enzymes, and then you have particular concentration of hydrogen ions for this metabolic reaction to take place. As well, you need to have a specific temperature so that you can have all these metabolic pathways working. These metabolic pathways, sometimes they have key steps in which you have an enzyme, and then if this enzyme is broken, you don't continue this metabolic pathway. And something that it can happen is that you will have buildup of a toxic molecule, and then that can lead to problems. Now, enzymes, just as most of the proteins, are basically coded in our genes, and when someone has defects in those genes, then the person will have a problem in metabolism. So now let's look at what are the components of enzymes. We're going to see how enzymes work, what are the factors that influence this work of these enzymes, and we're going to see what is competitive versus non-competitive inhibition, and we're going to see what are ribozymes. So enzymes and chemical reactions. So enzymes are catalysts. They speed up chemical reactions. Chemical reactions themselves, most of the times they will happen, but without an enzyme. But if you don't have an enzyme, it will take very long for this chemical reaction to take place. So these enzymes then, what they do is help by bringing these two molecules together that are the substrate molecules, or two or more molecules, and then that brings them closer. And with this physical closeness, you will have this chemical reaction much easier taking place, in which you can have either sharing of electrons or transfer of electrons, etc. All these chemical reactions. Now, the key work of these enzymes is actually to reduce the activation energy. So, here is an illustration of a chemical reaction between these two substrates, these reactants, A and B. So A and B are bonded by a chemical bond, whichever it is. So here in this x-axis, it shows the initial energy level and the final energy level. So as as time passes, these two molecules will be broken down into A and B products. But without an enzyme, which is shown with this blue line, the activation energy to break down the link between them, it is high. So then you will end up ultimately with A and B, but with these enzymes, what you do is you reduce this activation energy. And then that speeds up the rate of this chemical reaction. So, enzymes, when they combine with the reactants, form an enzyme substrate complex. And then this complex is going to be transformed and rearranged into products. Now, enzymes, they remain unchanged, and they will only react with these substrates. So substrates, they enter into a specific area of the enzyme that is called the active site. So this enzyme has these little gaps that specifically fit into these, well, interact with these substrates. So here is an enzyme with this empty active site, and then once it binds to these reactants, it's going to form the enzyme substrate complex. And then it can break down or rearrange these molecules within this substrate, and then you have some products that are released. So another place that the enzyme has, it is this place in here that is called the allosteric space or site. So sometimes some molecules can bind into this allosteric site, and then it will change the shape of the enzyme, and the enzyme, it won't be able to interact with this molecule. So this fitting specifically is called that key and lock mechanism. So here is another way to look at it. Some of them, they will have like these tiny gaps, and then the substrate, when it enters, it makes this enzyme to change a little bit so that it can perform its work to speed up this chemical reaction. So then, enzymes are specific. It's like if you're trying to open a door and you have the wrong key, I mean, it won't open. So that's the specificity. So with enzymes, then you can have 10 to the 8, 10 to the times 10 time faster reaction than without the enzyme. So there is a measurement of the rate that is called the turnover rate. So it's the number of substrate molecules an enzyme converts to a product per second. So it can be between 1 to 10,000 times, or it can be as high as 500,000. So then sometimes some enzymes are very efficient, other ones they are not as efficient, but they are very important. And then no matter if it's highly efficient or if it's not highly efficient, something that it can happen with enzymes is that if you have a lot of product, you will saturate them. It's like when someone goes to a famous restaurant that's very good. Of course, during lunchtime, that restaurant may have all the tables full, and then there will be people waiting to be seated. So that's the saturation. There's no seats for the persons to, or tables to sit on, and then you have to wait. So our enzymes also can saturate. Here are some common names of enzymes. Depending on what they do is how you call them. Typically, their suffix, the name, ends in ase. And we have oxidoreductase enzymes that participate in ox-redox reactions. We have transferase enzymes that basically transfer groups. Hydrolases that use water to break down chemical bonds, so they perform hydrolysis. Lyases, they remove atoms without hydrolysis or hydrolysis. Isomerases, they rearrange the atoms. So for instance, the conversion of glucose to fructose in the glycolytic cycle, we have an isomerase enzyme. An isomer is a mirror image of one molecule. Ligase, join molecules, uses ATP. We have DNA ligases. When DNA is being elongated, you form in one of these strands that is called the ligand strand, little pieces, the Okazaki fragments. So then you join them together with these ligase enzymes. There is this enzyme, the sodium potassium ATPase, that basically it's found at the end of the electron transport chain. And then as these hydrogen molecules flow through, this enzyme grabs ADP and inorganic phosphate, gyrates and bring them together to form ATP. It's funny why it's called ATPase. It sounds like if it was breaking down ATP. But no, it's not. It's actually making ATP. One of the exceptions to the rules. So we have in enzymes some components. One is called the apoenzyme, which is the protein portion. These apoenzymes are inactive without the cofactor. What is a cofactor? Well, it's a non-protein component, and it can be a coenzyme, which is an organic cofactor, or it can be an inorganic molecule like magnesium, zinc, cobalt. But when it's organic, it's called coenzyme. When it's inorganic, it's called cofactor. So then, when the cofactor or the coenzyme binds to the apoenzyme, you form a holoenzyme, which is basically the complete enzyme, functionally speaking. So many of the times, the enzyme is there, numbers of enzymes are perfect, but if you don't add that cofactor, that coenzyme, the chemical reaction won't take place. Many people like volunteer work, of course, and it's very commendable. But if the person only volunteers and never receives some sort of retribution, money, I mean, how is the person going to keep living, working? So here is the components of the holoenzyme. So here's the apoenzyme, the protein portion is inactive. And in here we have a coenzyme. And then you have a holoenzyme that is ready to bind this substrate. So here we have some coenzymes. So we have NADH, and NAD plus, and NADP plus, FAD, and coenzyme A. So this one, nicotine adenine dinucleotide, carries hydrogen ions, same as for this, but this one is the same, but it has a phosphate group. Flavin adenine dinucleotide also carries hydrogen ions, and this one actually carries more hydrogen ions than this. So these are reduced forms of these molecules. And these are sometimes derived from vitamins like vitamin B12, cyanocobalamin. So what does the enzyme needs to work? And a specific temperature. So depending on the organism that this enzyme is located on and the metabolism of this organism, it is that temperature. In us, typically it's like 37 degrees Celsius. That's a perfect temperature for our enzymes because that's the body temperature, 98.6 Fahrenheit. pH varies a lot. So we have some enzymes that work in acidic environment, others that work in basic environment. So one of the ways then to control the activation of these enzymes is by working with this specific pH. Some apoenzymes, they require acidity in order to work. And some, they actually become inactivated due to basic pH. And then substrate concentration. If you don't provide enough substrate, well, the enzyme will work, but once the substrate is gone, it will stop working. If you overgive substrate, well, the enzyme will work at its pace, but you cannot accelerate this chemical reaction anymore unless, of course, you add more enzyme. And the presence of inhibitors. We have different types of inhibitors that can prevent this enzyme from working. So, something that can happen with enzymes or any other proteins is if you get very high temperature, you can damage these enzymes or these proteins. They will unfold, they will denature, and then you will lose the property because you don't have that gap anymore to grab the substrates. And then I told you this before, you can provide excess of substrate and then you can saturate the enzyme and you get the maximum rate. And there are curves that are made for measuring the saturation rate. So here is an image showing you like the type of proteins that we have, or the tertiary structure of enzymes. Like in this case, we have some alpha helices, some beta sheets, and this is like the natural shape of this enzyme. But if you subject this enzyme to high heat, you will denature it. So it opens up and is not longer functional. So here we have the graph showing you what happens to enzyme activity in the y-axis as you change the temperature of this enzyme, going from 10 degrees Celsius up to 50. So the greater that you increase the temperature, you will reach a particular peak where you have the maximum activity of this enzyme. So in this case, this will be 37.5 or so in here. And then as you go over this temperature, like if you reach 50, the activity, it's just like as if you have it at 10. Now, something that happens, like let's say you have here 0 activity and 0 in here. Well, maybe you're not making any products at these two types of temperature, 10 and 50 degrees Celsius. But this one, if you start gradually increasing the temperature, you can still reach the right temperature, 37 degrees Celsius, and you can still have products. Now, with this one, the enzyme won't be functional anymore. You were working in a lab, you had a water bath, you put your test tube with your reagents, including the enzyme, and then you didn't check the temperature of the water bath. So, I mean, you saw that it was 37.5 degrees Celsius in the display, then you went do something else, and then you return, and then you see that it's at 50. Guess what? You have to restart your work again. So you have to put again, you throw away that, you put your water, your your substrate, your enzyme, your buffer, okay? And then you have to make sure that that temperature is kept. Let's say that you were in a hurry, you put the test tube in the water bath, you forgot to turn it on. You go and check the temperature of the water. I mean, it can be room temperature, 23 degrees, 25 degrees Celsius. Well, your reaction will be here. And then you come back 10 to 15 minutes later, and then, oh, I forgot to turn on the water bath. You turn it on, and you reach 37.5. That's perfect. You can still work with that test tube. For the pH, again, similar as for the temperature, you have streams in which you don't have enzyme activity, but you have the right pH, 7. And sometimes within this range of 7 or 4 to 6 or 7, you will have like some enzymes, they work at a pH 7.4. Others at 6, they don't work. So the concentration of hydrogen ions is important. And then here, it is to show you what happens once you saturate your enzyme. So you start here, and then the enzymes start basically increasing the number of products, and then it reaches a peak, a plateau, and then it keeps going like that. As long as you keep substrate, and then the enzyme won't give you like a linear plot. So competitive inhibitors basically binds to the active site. preventing the substrate from binding into this active site, blocking. Many drugs work as competitor inhibitors. One way that I illustrate this is like you all guys sit in the same seat every time that you come to class, and that's normal. I mean, I'm like that too. I'm a creature of habit. But let's say that someone comes before you and sit in your seat. Well, you cannot sit there anymore. That's competitive inhibition. So here is this enzyme. This is the normal substrate that binds into this active site. And then this competitive inhibitor binds into this specific area. So this substrate cannot bind here anymore because, I mean, it's a physical interference. There is no way, no matter that the competitive inhibitor will be there. Examples of this will be this PAVA, which is a normal substrate for the metabolic reaction of forming folic acid in bacteria. But there is this drug, sulfanilamide, that it looks similar. Look, they have a ring structure. All that they bury is in this region in here. So this one has this amino group, it's an amide, so it has sulfur as well. So then when you give this drug to a person, a sulfa drug, the bacteria won't be able to form folic acid. And without folic acid, the bacteria will die. So this is a competitive inhibitor. It's similar, but it's not the same. Now, non-competitive inhibitors interact with other part of the enzyme, that is the allosteric site. So then, in this case, when this non-competitive inhibitor binds to this allosteric site, you will have what is called allosteric inhibition. So basically that changes the shape of the active site. And then the active site can no longer bind the substrate. And this can be reversible or irreversible depending on how strongly this inhibitor binds to this allosteric site. And similar for competitive inhibition, guys. That's reversible for sure. So you always sit in your place, in your chair, and then competitive inhibition. Let's say that this was your chair. I come, sit here. You cannot sit. That's competitive. Then non-competitive will be putting something like that. This is the allosteric site. This is the active site. You come and then you see that you're thinking, oh, possibly someone is there. So then you don't sit there anymore. How can you overcome? That's reversible. In competitive inhibition in real life, I don't recommend you to grab a person unless you're stronger. Black guy. So then look what happens here in this allosteric inhibition. So this is the allosteric site. So then the non-competitive inhibitor binds to it here, and then see how these two round or semi-round active sites, now it changes shape, so this won't fit there. Now we have a feedback inhibition in which once you form a product, the enzyme doesn't work anymore. Why? Because you already have whatever you wanted. So basically, the end product of a reaction allosterically inhibits the enzyme from an early pathway. And in a metabolic pathway, typically is the first enzyme. So these are like the key steps. So here we have an example of feedback inhibition. So you have your substrate. Your substrate, it is bound, forming into the active site, forming the enzyme substrate complex. And then you have this allosteric site that first, it is not bound by anything. Because this is like, let's say this is like the first substrate that enters into this metabolic pathway. And then out of this, you form an intermediate A. And then you have a second enzyme that is going to convert this intermediate A into an intermediate B. And this intermediate B is going to be converted by this enzyme, number 3, into the end product. So then this end product is going to accumulate and later on is going to bind into this allosteric site of this enzyme 1, so that this enzyme 1 doesn't produce any more of these intermediates. And see how enzyme 2 and enzyme 3, they don't have these allosteric sites. So then indirectly, another way to inactivate an enzyme is if you know your product, you can add it into this metabolic reaction, and then you can inactivate this enzyme by binding it to this allosteric site. Now, ribozymes. So ribozymes are RNA that functions as catalysts by binding substrates and act upon them. And just like as before, for the enzymes, they're not used in the reaction. And the cells frequently use them to cut and splice RNA. And then they are also involved in making proteins in ribosomes. Basically, they're found in ribosomes. That's why they're called ribozymes. Okay, what is a coenzyme?
It's an organic molecule.
That does what?
It's an organic non-protein that helps build, it's the second piece to an enzyme that makes the enzyme actually active.
Mm-hmm. Why is enzyme specificity important?
substrate, and it's not going to react with a bunch of other substrates. It has to be specific to react with that substrate.
Imagine if instead of having 20, 30 enzymes in our body, we only have only one enzyme that does all the metabolic reactions in our body. I mean, why wasting 29 enzymes? Why not have only a single one? Yes. And not only that, let's say like out of that one, you have a lot of numbers to do the same. But what will happen if someone have a genetic defect that prevents that enzyme to form? Then you will die, right? So that's why we have redundancy, but at the same time, a specificity. So what will happen if we have less than optimal temperature with the enzymatic reactions? Will they occur?
Much lower rate.
Yeah, a much lower rate, or maybe not. But what about if the enzyme denatures? Can that happen at all? No, not at all. So then, what is the difference between competitive and non-competitive inhibition?
Competitive binds to the active site, whereas non-competitive has the allosteric site, which isn't the active site.
Mm-hmm. And what is a ribosome? Mm-hmm. You can splice the RNA. Now let's look at specific chemical reactions now, oxidation, reduction, and phosphorylation types, and some metabolic pathways. So, for producing energy, the molecules in our nutrients have these electrons that can form bonds between atoms. And then we can break down these molecules by making them much easier for they to go into these metabolic pathways, like in the case of glucose, and then they can go and fit into these larger metabolic pathways, like Krebs cycle, oxidative phosphorylation, that can generate more ATP. So some of these catabolic reactions basically concentrate the energy by forming bonds of ATP. And each of these bonds of phosphate groups, when they are broken, can release energy. Now, in ox-redox reactions, you have removal of electrons in oxidation and acceptance or gaining of electrons in reduction. They are coupled. You have to have a molecule that is oxidized while the other one is reduced. So there is this mnemonic oil rig. Basically, oxidation is loss of electrons, O, L, and then rig. Reduction is the gain of electrons. And then Leo loss of electrons is oxidation and gain of electrons is reduction. So two ways to memorize it. So here's the coupling of oxidation and reduction. So here we have A and B molecules. So this one has an electron that is going to transfer the electron to B. So since this one is losing electrons and is transferring to B, A is going to be oxidized and B is going to be reduced. And there is this transfer between the state of oxidation and reduction. So B is reduced and becomes, is oxidized and becomes reduced, and A is reduced and become oxidized. So that's why both of them together, they are called ox-redox or redox. Okay. So in biological systems, because this can happen even like with metal, steel can get oxidized, of course. But in biological systems, electrons and protons at the same time are removed, and this basically it's equivalent to forming a hydrogen atom when you have hydrogen atoms interacting with NAD, FAD, any of these molecules. So in this case, these reactions are also known as dehydrogenations, biological oxidations. Like when NADH, it basically brings these electrons into the electron transport chain, these will become dehydrogenated because it's, instead of being NADH, once it releases these electrons, it's going to be NAD. So it's going to become reduced. So I get ahead of myself. So here we have this organic molecule that can transfer, in this case, 2 electrons to this carrier, NAD+. So this is a coenzyme. It's an electron carrier. So it's getting oxidized. And then the organic molecule, once it's oxidized, is going to reduce this carrier. So now this one is going to be called NADH plus because it's going to have a proton, hydrogen. And then this proton is going to go into in the case where the cell might have an electron transport chain, it will go into electron transport chain, and then you will transfer these electrons into these proteins within the electron transport chain, and this one that it was reduced, NADH plus, is going to become NAD plus, so it's going to be reduced. So for generation of ATP, it happens through phosphorylation. So basically, ADP will grab an inorganic phosphate, and this process is going to be called phosphorylation. And we have a specific enzyme for this, like the ATPase synthase. ATP synthase. So you can have AMP, but you can have ADP in the case of in glycolysis. you will have ADP, so you have adenosine base with two phosphate groups attached to them, and then with energy and in an organic phosphate, you end up with ATP. So that's one type of phosphorylation, forming ATP. And then we have something that's called substrate level phosphorylation. In this case, ATP is generated when you have a high energy phosphate group that is directly transferred from a compound to ADP. So in this case, this will be the compound that has this phosphate group that can add it into this ADP, and then you end up with a dephosphorylated compound with a phosphorylated ADP that now we call it ATP. So either you can have free inorganic phosphate in the cytoplasm of the cells, and then you add it to ATP. In that case, that's phosphorylation. But if this inorganic phosphate is attached to a compound, in this case, it's substrate level phosphorylation. And then in oxidative phosphorylation, well, you can still generate this ATP, but in this case, it is part of the electron transport chain, or ETC. So these hydrogens that come with FADH or NADH that gets into this system of proteins that we have in the inner membrane of the mitochondria, these hydrogens will be transferred from the matrix of the mitochondria into the intermembrane space. And you will form something that is called chemiosmosis. And that basically is the transfer of the hydrogen ions through tunnels in the ATPase synthase, and that is going to basically, as these hydrogens are returning to the matrix, going to move the subunits of this enzyme, which grabs the ADP and inorganic phosphate, and then you will basically form ATP. And in this electron transport chain is where we form most of the ATP rather than the other two types of phosphorylation. So everything is just, again, carrying hydrogen molecule from NADH or FADH plus, which will be reduced to NAD and FAD. And as they release these electrons into this chain, these electrons will flow, and then they will accept the oxygen, which is the last acceptor of electron, will accept the electrons, you form water, and then you will have this transfer of hydrogens from the inner membrane space into the matrix, and then you will have the formation of ATP by this ATPase enzyme. There is a special type of phosphorylation that's called photophosphorylation because it is phosphorylation with the use of light. And it happens in the chloroplast. So you have these pigments, the chlorophylls, that trap light, and that drives the transfer of electrons, phosphorylation, from the chlorophyll into carrier molecules that ultimately going to form ATP. So it's similar to the oxidative phosphorylation, but in this case, you have to have light to activate chlorophyll pigment. So here is the cyclic phosphorylation. So you start here with a ray of light hitting this chlorophyll 2 pigment, and that is going to lead to the basically release of 2 electrons that are going to go into this chain of proteins within this chloroplast and then you will have an electron carrier here. And then that is going to hit back this chlorophyll, and then you start a chain reaction that is going to basically increase the levels of energy, release of inorganic phosphate, and that inorganic phosphate is going to help for ADP to bind to ATP, sorry, to inorganic phosphate, then you form ATP. So we have photosystem 1 and 2 in this case. So what are the three ways that you form ATP? Or even. Mm-hmm. Phosphorylation itself, photophosphorylation, when you have a compound with phosphate, inorganic phosphate, and then you give it to ATP, what is it? Substrate level phosphorylation. And then you have the oxidative phosphorylation. So now let's look into some metabolic pathways of energy production. So the metabolic pathways, so they are series of enzymatically catalyzed chemical reactions that happens in a sequence. And that helps to take away energy from organic compounds to store it in its chemical form, ATP. So here we have a hypothetical metabolic pathway. So your starting material is A, and then you're going to you're going to take away from A some hydrogen molecules. And then as you are removing these hydrogen molecules from A, you will have a carrier of these hydrogen molecules, these electrons. So you have NAD that it was reduced, and now it's going to become, that it was oxidized, now it's going to be reduced. So then you will have the conversion of A into B as a starting material. Now B, since it's going to have inorganic phosphate, so then you will have the formation of ATP when you have ADP in contact with B. So this is basically what you will call like the substrate level phosphorylation. Now you will end up with C product that now can be converted into D, or you can reverse D into C. Now in this case, when you convert C into D, and then you add oxygen, you will have this D molecule entering into a series of steps that ultimately will convert this D into E, and at the same time, you will be generating CO2 and water. What you're looking here, it is actually what happens in glycolysis and also in the Krebs cycle. So yes, we have different enzymes for these metabolic pathways, But what is the main purpose of all of these metabolic pathways with the different enzymes?
To extract the energy and store it as ATP.
Extract energy and store it as ATP. And then with that energy, you can drive more metabolic reactions. Like, for instance, if you want to make proteins, if you want to make nucleic acids, if you want to build a cell, you have to have these metabolic pathways. So now we're going to see glycolysis specifically. We're going to see Krebs cycle, we're going to see oxidative phosphorylation, and we're going to see fermentation. So most organisms use glucose as a form of energy, or carbohydrates, I should say. And glucose is very common as an energy source, and glucose can be used as by two ways, or carbohydrates, by cellular respiration and fermentation. Now, cellular respiration includes glycolysis, Krebs cycle, and the electron transfer chain, GKE. Cells, in order to survive, they have substrates available, like carbohydrates, they have lipids, they have proteins, and they have nucleic acids. Nucleic acids as a whole, typically they are not used by animal cells as a source of energy. But yes, we can use lipids, we can use carbohydrates and proteins. Chemically speaking, glucose molecules are very simple. They're not as complex as proteins or as lipids. So that's why the number one source of energy that animal cells prefer to use is glucose. Second will be lipids. Third will be proteins. The reason for this, it is because proteins are so precious. They form channels, they form receptors, they form cytoskeletal proteins, very important enzymes that are very important, so that's why we spare them as much as possible. Sometimes some people, they go into these diets, and yeah, they start losing weight, but it's not weight in lipids, it's weight in proteins, and that's not good. You're losing your muscles. So then here we have the overview of respiration on the left side and fermentation. Fermentation as a whole is shorter than respiration. In respiration, basically your glucose is converted into pyruvic acid, then acetyl-CoA goes to the Krebs cycle, oxidative phosphorylation, and you generate a lot of ATP. In fermentation, that pyruvic acid or any other derivative will form end products like acids and CO2, but without undergoing all these sequence of steps in the Krebs cycle. So, in glycolysis, you oxidize your glucose molecule to pyruvic acid or pyruvate, and then you produce 2 ATPs and 1 NADH. This is the first step of carbohydrate catabolism, and another name of glycolysis is Embden-Meyerhof pathway. It has 10 reactions catalyzed by different enzymes. So you have a preparation stage. You add, the first one, add ATP into glucose, so you phosphorylate glucose, glucose 6-phosphate, you end up with glucose 6-phosphate. And the reason for this is because you want to bulk glucose molecule. That will prevent glucose from exiting the cells. And not only that, because it's a bulk molecule with ATP, but when you add this, when you phosphorylate your glucose, you add a negative charge. And since the membranes internally are negatively charged, then by the glucose molecules getting close to the membrane, they're going to be rejected by these opposite effects. So negative charges to negative charges repel. So you physically and chemically trap your glucose. And then glucose will be split into two molecules. One is going to be glyceraldehyde 3-phosphate, and the other one, the hydroxytone phosphate. And then this molecule is going to be converted, the hydroxytone phosphate is going to be converted back into glyceraldehyde 3-phosphate. So then since you have these phosphate groups in glyceraldehyde phosphate, they can enter into the remaining of the reactions. So here is the initial steps. You phosphorylate your glucose. You end up with glucose 6-phosphate. And this glucose 6-phosphate is going to be now isomerized into fructose 6-phosphate. And then fructose 6-phosphate is going to be phosphorylated, and then you end up with 1,6 fructose. And then this fructose is going to split, and then you will have your the hydroxyacetone phosphate, and then you have a 3-glyceraldehyde phosphate. And this one, the hydroxyacetone phosphate is going to be converted into this glyceraldehyde 3-phosphate. So this is the first steps. Now, the next segment is going to be the energy conserving stage. So your 2-glyceraldehyde 3-phosphate molecules will be converted into 2-pyruvic acid. So they will be oxidated. So in this step, you will generate 4 ATPs and then 2 NADHs. Glycolysis happens in the mitochondria, in the cytosol. Krebs cycle in the mitochondria. So since NADH is going to be a carrier of hydrogen, it has to be translocated into the mitochondria. So over there, you will have generation of ATP additionally with the NADH that it is formed during the Krebs cycle. So in glycolysis, you have these 2 ATPs that are spent, and then you generate 4. So you end up with a net gain of 2 ATPs. And then you have the pyruvic acids as the end product, but you have as well this NADH and 2 hydrogen molecules. So overall, the net gain will be 2 ATP molecules for each of molecule of glucose that is oxidized. That's very little. So then you have this pyruvic acid that is going to enter in the glycolytic cycle, in the Krebs cycle. So that's all